How was? I really want my.
Zoom.
That's fine. Oh. Yeah. Oh, man, you're right. Oh, that's right. Yeah, it's actually one thing.
Boop, boop.
And my That's what
Yeah. Because they're just like, I don't. I know, I had an issue till next year, and then I'm still coming, if I'm just more this. I do great. I feel like I'm gonna start sending questions and I was like, pretty aggressive.
Oh no.
Yeah, it's like I think there's a lot of like the questions, and I was like, oh, I. Yeah, so it's like.
Ugh.
Oh.
Okay, you guys ready to get started? Woo! Okay. Okay. So where we're at, where we're going, all that fun stuff. How did the exam feel to everybody? Like a valid assessment of the information that we covered?
Yeah.
Okay. I mean, honestly, if you ever sit through one of my exams and you're just like, walk out of the room and be like, what the hell was that? always feel free to send me an email after. That's very valid. Just because I very much try to make sure there's an alignment between what I present to you in class, what I confer to you as are my expectations, and then what shows up on the exam. I have no desire to trick you or do any of that other crap that sometimes faculty do. Let's be honest. Okay. So with that being said, we are in week 11 of the semester. Week 11, man, we are 2/3 of the way through the semester. It's kind of wild. And we're coming up into November, which is going to be one of those kind of crazy months where we have a day off here and a couple days off there, which actually does impact this class because we won't have class next Tuesday, and then we won't have class obviously the Thursday of Thanksgiving. So hopefully you have that in your calendars. But there's a couple things I wanted to attend to today, the big picture of this class before we dig into like the material. So I guess first thing I wanted to point out here is under week 11, I posted kind of a fun thing possibly for you to check out. This is a student edition of the career services, basically section of our campus. If you are interested in, let's say, things like public service or what have you, this is just a newsletter that, our career services group puts out. There's a grad fair coming up, for example. Maybe that's something that you're interested in. And so, yeah, if this is stuff that you are possibly interested, long-term career trajectory types of information, I've posted it at the top of week 11, so feel free to check that out. I'm a career champion, apparently, because I work with career services. So I'll continue to give you guys tools to just apply as you see fit. It's not required for this particular course, but I figure information is power. So if you're interested, check it out. If you're not, then don't. Cool. So moving forward, the other thing I kind of want to draw to your attention is the final project, right? So the final project is going to be presented in class, our last week of class. I have a strong feeling this is something a lot of you have not necessarily thought about yet because I checked our sign-up sheet. We have one person who signed up. So we're going to take a little bit of time to kind of discuss exactly what this final project is and what it looks like and kind of what my expectations are. Okay, so this is the written document in terms of the assignment directions, but I'll just kind of give you my elevator pitch for a set assignment. You will have 15 minutes to present as a single person, as a group. I don't care how big your group is. I don't care how small your group is. If you're the kind of person who flourishes by yourself, so be it. So what you will first do is select a topic in neuroscience. Again, I don't really care what that topic is. It can be anything. If your passion is aphantasia, everybody can Google what that is. You can do this project on said thing. This is an opportunity for you to focus on what you're really passionate about in neuroscience, okay? Now, with that being said, going into the final, it would be awesome if the topic that you chose is something that we've covered this semester because, hey, maybe your project will help us study for the final. So keep that in mind as you're choosing your topic. But like I said, if your topic is not something we've even touched upon in this class, totally fine. I just kind of love seeing where your creativity is going to take you. You're going to develop content for your presentation. So like I said, you've got about 15 minutes for said presentation. And what I want you to do is, I know that so often we are so used to standing up here. When I say presentation, you're like, I'm going to sit up here with my slide deck and present for 15 minutes. That's actually not what I want you to do. What I want you to do is figure out an interesting / fun, active way you can engage your audience to kind of demonstrate or discuss or interact with the material that you're presenting them. Okay, so again, what I'd like you to do is kind of introduce what we call active learning into the classroom related to your topic. So usually the way this looks is we'll have kind of maybe like 5 minutes, maybe 4 or 5 slides that introduce your topic. You start to present a little bit of material to your audience. Maybe you present them with a problem, with a question, and you ask them to kind of dig into that material and do some type of like active participation. We've done a little bit in this class. We've done some website work. Wouldn't necessarily like lead on that, but any type of like active engagement exercise that you can do. I've incorporated a link here about active learning strategies. Again, try to think about something that's outside of the box that you've really enjoyed in a class and maybe apply it to your particular topic. Okay, Now, I know this is kind of a weird assessment. You probably haven't done this for any other classes, so I'm going to pause now and ask if anybody has any questions. Okay. I will be asking you to use visual aids. Again, the PowerPoint is usually the kind of go-to thing here. You've got 15 minutes, again, at the max 4 to 5 slides. Okay. Practice time management. Make sure during that kind of active component of it that you give your audience enough time to interact with said material, and that you interact with your audience through said material. Okay. So as you're presenting this, as you're asking your audience to do said activity, you want to be checking in with your audience, making sure they understand the directions, making sure they understand what they're trying to do. Be pretty directed in terms of what you're trying to accomplish during that time. You'll practice presentation skills. If you have any supportive materials, so be it. I mean, do you guys want to see an example of this? Okay, I won't do it today, but I've got an example that happened, I think, like 2 or 3 years ago. And it was actually pretty cool. It was a pharmacology example of like receptor binding. And the student's still in one of my other classes, so I'm going to ask him to give me his materials. But we can do an example of this, which actually will lend itself to some of the material we're covering this week in Thursday's lecture. Okay, so we'll do an example on Thursday so you can kind of see what the expectations are there. I've posted these directions, which I kind of verbally just gave you a little bit of that. There's also the grading rubric if you want to see how you're evaluated. First off, if you just show up that day, you automatically get 20 points. And then all of the rest of the points is based off of kind of audience engagement, use of visual aids, clarity of presentation, content knowledge, time management, and your overall presentation skills. Basically, I'm asking you to come prepared and just clearly present your ideas, okay? So like I said, we'll go over an example on Thursday. Hopefully that'll maybe start getting your thought patterns stimulating. Okay, so now what I'm going to do is give you all, we'll say like 3 to 5 minutes to maybe go touch base with somebody that you have maybe previously talked to about working on a project with, or if you haven't attended to the sign up sheet yet, you can sign up on the sign up sheet about the particular topic that you've been discussing, just because I kind of want to make sure we get something on paper today as to what you're discussing. And if you're working alone and you just want an opportunity to chat with your neighbors, feel free to do that too. So like I said, I'm going to leave it unstructured now, 3 to 5 minutes. Maybe quickly bounce off some ideas of one another, hit the sign up sheet, sign up, and we'll kind of come back to your regularly scheduled class after that 3 to 5 minutes. Okay, any questions? No? Okay, get to it. 3 to 5 minutes and try to identify what you're going to be doing. Anything close, sir? I didn't see it. It's okay. It'll be fine. Yes, I was like, I know, when I heard about this, I knew exactly what it was. Had a great. You have a. Yeah, I already have it. It's written down. That's okay. I'm good. Okay, cool. Very excited about this. Yay! Yeah. Sometimes it's so minimal, so I'm like, we didn't do one. Okay. I know what I'm saying. I've been cooking. I've been cooking.
And then. Yeah. I was thinking about, let me see how much time I actually have. I might have to send out maybe other. Yeah, sure. But I do find it for that kind of stuff.
Okay, I'm gonna give you about 2 more minutes. We'll start kind of regularly scheduled class at 2:15. So if you haven't jumped on here yet, or if you have, again, realize that what you write here is not a commitment of any sort. Like, then you've got a plan, so go ahead and write it down. You can modify it later if you go in a different direction.
Yeah.
Mitchell offered you to like work research.
He offered to do like crop dog. My friend's guy.
Oh, that's the if I like pick up.
See if I let.
I don't know. I don't know about when.
Okay.
I got like,
See, like my.
come with the suit and tie next time.
Yeah.
You can't.
I very much held expect if they have the street. Is this linked to like Levin or is it in the like?
It's at the very, very
It's
bottom.
like, okay. It's like-
So,
Like the notes, like we like.
Yeah.
Do this in January.
the
Like-
final project section at the very bottom. Final project sign-up sheet. Okay. What? Oof.
5:20.
Oh, on what day?
5:20?
Yeah, December 9th.
Sure, I don't know. I don't know. I haven't looked up.
Yeah.
Great.
Don't be afraid. You have to realize that this is basically an archaeological dig of this entire course over the last 5 years. So it's possible you saw something that didn't apply to this year. So don't stress about it.
It's like Friday. I'm sorry.
Okay, so I planted the seed. Maybe you've been able to have some conversations. We'll do an example on Thursday, and then hopefully that'll kind of stimulate your creative minds. And if you have additional questions, we can kind of answer or attend to them at that point in time. If at any point you're working your way and you're kind of preparing your project and your presentation for that final week, and you even just want to meet one-on-one or 1-on-3 or whatever your group number is, to just kind of talk more in depth about said project, I'm happy to do so. So just shoot me an email, and we can get something on the books so you can kind of get some feedback as to what you're preparing. Okay, so now let's shift over into like this week's content. So this week is probably the week that you're just like, yes, this is why I came to a neuroscience or registered for a neuroscience class, is we are actually going to talk about neurotransmitters, which is kind of exciting. As we talk about neurotransmitters and we've discussed the 5 steps of neurotransmission, hopefully everybody feels comfortable with the 5 steps of neurotransmission. And now we're going to apply it to all of the individual neurotransmitter systems. So here on our left column here, this is at the very bottom of week 11. This is not required for you to turn in. This is just a worksheet that helps, hopefully aids you in conceptualizing what we're going to be talking about in terms of content. So now today we are going to cover glutamate, GABA, and actually acetylcholine. You might even want to modulate this. This is a PowerPoint slide and a table, in which we'll be talking about the biosynthesis, the storage, the release, the postsynaptic receptors, and kind of the steps. The termination, I think, is also down here at the bottom. Sorry, I have to drive kind of backwards. Yeah, and then you have signal termination. So again, if this is the way your brain works, if you're a table person, I'm obviously very clearly a table person. You don't have to be, but whatever makes kind of the most sense to you in terms of organizing this information. please do so. And yeah, I don't know, like I said, I like tables. Okay, so this is it. We're getting into the neurotransmitters. We're getting into what the neurotransmitters do, what their structures are. We've kind of dabbled around them the last couple weeks, but this is literally step by step through each of the different neurotransmitter systems. So today, our roadmap is, we're going to talk about the 5 steps of neurotransmission for glutamate, GABA, and acetylcholine. In addition to that, we'll also talk about physiological effects, some of the receptor subtypes for said neurotransmitters, and then we'll finish up with some interesting clinical information. Again, maybe I'm using the term interesting too loosely here, but we will just talk about some neuropharmacology related to that neurotransmitter system. Okay, so again, very big picture here. Sometimes when we talk about topics, it's hard to conceptualize them because they seem kind of sporadic and independent, and it can be useful to see the relatedness of neurotransmitters to one another. So based off of their structures alone, we can see here we have glutamate and GABA that are basically just modified amino acids. We'll talk about what their precursor molecules are. We'll talk about how they get modified into their final signaling form. Acetylcholine is interesting. It's an ester. That's its chemical structure. It's composed of acetyl and CoA, and we'll talk about that process when we get to that section. We have serotonin, which is actually a monoamine based off of its structure, and we can see a little bit of the biosynthetic pathway here. And then we have our catecholamines, and again, dopamine would be one of the predominant catecholamines that we'll discuss in terms of neurotransmission, but I'm just going to quickly point out here, we also have norepinephrine and epinephrine. Are those important? Why? Are norepinephrine and epinephrine important, and why are they important? They're like, we didn't cover this yet. Why are you asking us this question? Okay, so norepinephrine and epinephrine are kind of the primary signaling molecules for your autonomic nervous system, okay? They bind to our alpha and beta adrenergic receptors that we do have expressed in our brain, but also are expressed very heavily in our physiology across our bodily systems, right? Your respiration rate, your heart rate, your, again,
You
your
can't be learning and looking at your phone at the same time.
all is going to be modulated.
Literally try. You're crippling yourself, not using it.
Sorry.
Did it.
So those molecules,
Yeah.
those
She
neurotransmitters
explains
are
things
important
really well.
because they operate primarily in
This
our
is the first
autonomic
time I'm learning
system
this.
and not our central
I
nervous
have a
system.
little more
I'm going
background.
into them a ton this
A little
semester.
more.
I'm happy to talk about them at any length if you have questions about them. Okay, so again, this is kind of how our neurotransmitters are related to one another structurally, aka chemically. So let's first start by talking about our amino acid neurotransmitters. So our amino acid transmitters, as I introduced on the last slide, we have GABA and glycine and then glutamate. GABA and glycine are going to be our inhibitory neurotransmitters. Thus, I put them in red. Our GABA or glutamate is going to be our primary excitatory neurotransmitter, and I put that in green. Okay, inhibitory slows down our neurological systems. Excitatory is going to promote excitation of the systems. Our GABA and glycine are released by generally interneurons. We're going to see glycine being our neurotransmitter that's operating a lot in our spinal cord, and then GABA itself is kind of ubiquitously found across our entire kind of cerebral cortex. I love these pathways here. It literally just looks like somebody got wasted and was just like, I'm going to draw on this brain, but I think it's probably fair enough to say that GABA and glutamate are found all over our brains, okay? So I often think of these amino acid neurotransmitters as being kind of our primary drivers to both the cognitive effort that we make, as well as the depression that happens related to our neuronal signaling. And then our other neurotransmitter systems are generally just going to kind of tweak those a little bit. They're just going to tweak the signaling of both GABA and glutamate. Okay, so let's dig into glutamate first. So glutamate, like I said before, our primary excitatory neurotransmitter, and we're going to talk about kind of those 5 steps of neurotransmission related to glutamate. Okay, so here we have our 5 steps. Synthesis, storage, release, binding, and termination. Okay, hopefully this feels like a comfortable place for you. So now let's talk about what exactly happens for glutamate. So glutamate is synthesized from glutamine. Here we see glutamine coming in. If you're kind of paying attention here, you can see how this is kind of a recycling process that happens here. Glutamine is going to be our precursor molecule that's going to be converted into glutamate by an enzyme called glutaminase. Okay. And again, I know this feels kind of biochemistry-y, but it is important to know some of these enzymes because some of these enzymes can be pharmacological targets, or they can be ways in which we can tell one neuron from another, which we'll talk about when we get to GABA. So you have glutamate that is being produced in your presynaptic terminal. It gets loaded into your synaptic vesicle via specific protein embedded in that vesicle called V-glut. V-glut is your synaptic vesicle transporter for glutamate. You know, science doesn't always help us out with nomenclature, but this, for the most part, is pretty straightforward, right? Glutamate becomes glutamine, glutamine becomes glutamate through glutaminase, and then V-glut is going to be our vesicular transporter for glutamate. And then upon the arrival of an action potential, you have release of glutamate at the synapse, and that is going to bind to these different receptors, okay? You'll notice that these receptors are different from one another. What type of receptor is this? Structurally, is it ionotropic or G protein coupled? This is a G protein coupled receptor. We know that because it has 7 transmembrane domains. This subtype of receptor is called NEURs, which stands for metabotropic glutamate receptor. Again, pretty intuitive. And then we have our 2 different ionotropic glutamate receptors. We have our AMPA receptor and our NMDA receptor. Those receptors are based off, those receptor names are based off of the molecules that were originally used to characterize those receptor subtypes. So don't get too down in the weeds, but we will talk specifically about how AMPA receptors and NMDA receptors differ from one another. They kind of do different things. They kind of signal in different ways. The signal termination for glutamate is going to be reuptake through transporters expressed on both the neurons as well as the astrocytes, although the astrocytes tend to have a much higher level of expression, and those are taken up through excitatory amino acid transporters, also called EATs. Again, EATs stands for excitatory amino acid transporters. Now, in the glia, aka the astrocytes, you are going to see glutamate get turned into glutamine through glutamine synthetase, or synthesizing glutamine. And then that glutamine gets released from astrocytes and can be taken back up into the presynaptic terminal, and the whole process can start all over again. Okay? So these are all steps in the neurotransmission of glutamate, the primary excitatory neurotransmitter for our nervous system. So not a trivial thing, right? Like every single thought that you're having right now, this is what's happening in those cells. You are literally releasing glutamate and it's binding to your AMPA and NMDA receptors. Like this is happening in your brain at this very moment. So let's talk a little bit more about our receptors, okay? So it acts through both ionotropic receptors. Again, we have, and this is NMDA, AMPA receptors. We also have kainate receptors, which we don't discuss a ton in this class, but definitely still have a function. And then you have your metabotropic receptors. Again, slower neurotransmission. They're G protein coupled. And we have 8 different kind of varieties or subtypes of mGluRs that facilitate that action. Okay, so like I said earlier at the beginning of class, I love myself a table, and here we go. So this table here, you can see on the far left, you have the family, whether it be ionotropic or metabotropic receptor, and then we have the subtypes of said receptors. So again, if you're walking your way through this, you're like, I got it, AMPA, NMDA, mGluRs, no problem. It's a lot more complicated than that. I will just, I'm not showing you this to overwhelm you, but to just give you some appreciation for the nuance that our physiological system actually has. So based off of subtype of receptor composition can determine the properties of that receptor. That's why we even care about subtypes more often than not, is because based off of the receptor composition, it's going to determine whether that ionotropic receptor just allows sodium to pass through, or if it allows sodium and calcium to pass through. And calcium is a pretty important intracellular signaling molecule. So again, that's one of the reasons we spend so much time trying to figure out these different receptor subtypes. And then again, to take it to more of a clinical place, like how to pharmacologically modulate them. So for this, you can see we have drugs that we've developed as agonists, which are going to activate said receptor, and we have antagonists, which are going to block each of those receptors. So you can see here a set of drugs that kind of do these jobs, whether it be activate or block. Most of these drugs are not used clinically. More often than not, they're just used in the benchtop research to understand, again, kind of the downstream physiological functions of each independent receptor subtype. Does that make sense? So a lot of these drugs are just more tools. They're not necessarily something that would get prescribed at a pharmacy. Okay, if we move down to the metabotropic, we can see that we do in fact have 8 different subtypes of mGluRs, but they're grouped in 3 different ways. And that grouping, for the most part, is based off of kind of the way that they're coupled to their downstream G proteins. So our group 1 mGluRs are going to be G alpha Q coupled, meaning that they have kind of, again, more excitatory action. And then you have some G alpha I and O metabotropic receptors that are coupled to G alpha I and O, that tend to be a little bit more inhibitory in action. Again, if we don't get too much down in the weeds there. Okay, ready. NMDA receptors. This is something that will be on your next exam, okay? NMDA receptors are unique receptors because they are what we call coincidence detectors. And when I say coincidence detectors, it means that multiple things have to happen for these receptors to signal, okay? The 3 things that have to happen for an NMDA receptor to signal are, and again, this is usually where the question comes into play, is first, they are glutamate receptors, so they have to bind glutamate, okay? So for them to become activated, they have to bind glutamate. Number 2, the second thing that has to happen is they have to bind glycine. Glycine, if you remember a few seconds ago, is an inhibitory receptor subtype, but glycine is required for NMDA receptors to be active. So they have a cofactor here of having to bind glycine. Third thing is that they have to have some type of depolarization event that dislodges the magnesium block from the NMDA receptor, okay? So NMDA receptors usually have a magnesium block. Oh, I think it's supposed to be good. The magnesium block just hanging out here. It's almost like you have a clog factor. What is magnesium charge? What is it? Is it positive or negative? It's positive, okay? So when the inside of the cell becomes more positive, it causes that positively charged magnesium to pop off, okay? So it's a very effective coincidence detector via the presence of glutamate and the depolarization of that cell. It's kind of like the way the cell checks that something's really happening here. Okay, so that's how NMDA receptors work. Other important factors here, they're heteromers. There you've got all of your NMDA receptors are going to have a gluN1 subunit, and then a different varying concentration of gluN2 receptor subtypes. The minimal receptor composition is of the gluN1 and gluN2 dimer. They are highly permeable to calcium. Calcium is a very important intracellular signaling molecule. Calcium is one of those things that leads to other events like long-term potentiation, which is our molecular mechanism for learning and memory. It is pretty well characterized that there are several different types of long-term potentiation that are mediated specifically by the NMDA receptor because of this influx of calcium that it can facilitate, okay? So learning and memory happens when you have a depolarization event, the binding of glutamate and glycine binding. Now keep in mind, I'm mentioning glycine binding here intermittently, but glycine is almost ubiquitously found in the brain, so you really don't have to worry about it being released by a secondary source, if that makes sense. So glycine's kind of an old factor for the most part, but it's important to measure that there's kind of these 3 things that have to happen. Okay, so how does a cell get depolarized? For a cell to undergo depolarization, what is the most common way that happens? Maybe we need to step back. What is depolarization? Depolarization. Okay. Polarization. Okay. Depolarization is when the cell becomes more positive. How does the cell become more positive? With an influx of usually sodium, right? Okay, so I'm telling you here that NMDA receptors are only kind of active if there is a depolarization event. For a depolarization event to happen, you have to have an influx of sodium occur. We are all in agreement with that, right? Well, what other receptor subtype binds glutamate and allows sodium to come in? Our AMPA receptor, okay? So at a glutamatergic synapse, usually you will have co-expression of both an AMPA receptor and an NMDA receptor. And what happens is that glutamate binds to both of those said receptors, AMPA and NMDA. What happens is AMPA receptors immediately open and cause an influx of sodium. So then you get your depolarization. And then the secondary event that happens is that magnesium block comes off of that NMDA receptor that is also bound to glutamate. And that's what allows both of these receptors to kind of both signal at the same time. Okay, but these receptors have different kinetics, meaning that AMPA receptors tend to open up really quickly and desensitize really quickly, which kind of sounds a little bit like our voltage-gated sodium channel, right? And then NMDA receptors open very slowly and close very slowly. Again, not too dissimilar from what we saw in our voltage-gated potassium channels. So they have different kinetics, meaning the rate at which they open and close. And again, it feels like I'm harping on this a lot. Sorry, this is kind of, whenever you get into an area or field that somebody has studied, I apologize. We get crazy about this stuff. But again, this is again how both AMPA and NMDA receptors act with one another. Lastly, again, we have archaic acid, which are heterohomer tetramers. Tetramer meaning that there's 5 different subunits that require extracellular sodium and chloride for activation and do not exist kind of in the post-synapse signaling complex. So again, NMDA and AMPA receptors are kind of our 2 big ionotropic receptors. This is kind of, again, showing you what happens at rest. You've got, again, your presynaptic terminal, the release of glutamate, that magnesium block is present. And then during a postsynaptic depolarization, you have glutamate that is bound to both receptors, sodium's flexing in, causing the cell to depolarize, and that means magnesium block to kind of pop off, and it allows for calcium to flux through. Okay, so not a wild question for me to ask on next exam is like, how do AMPA and NMDA receptors work together at the glutamatergic synapse? Okay. All new information to you, but I'm going to give you a second. Pause, process, and then turn to somebody sitting next to you and share your description of how AMPA and NMDA receptors work together to allow for synaptic transmission. Okay, so come up with your own description of it, turn to somebody sitting next to you and have just like a quick conversation.
NMDA means that depolarization for the positive and negative response. And so then when there's the binds to the N receptor that allows the sodium and gives it then and then it can bind. Not usually you were saying. So. Because if I'm writing well, I mean, people already wrote that. I got both people and then yeah, it seems like that's
Okay, go ahead and find your seats. Okay, so we have our AMPA, we have our NMDA receptors. So let's continue to discuss a little bit more. Okay, so NMDA receptors. If NMDA receptors end up being your jam and you're like, these receptors are awesome, this is exactly what Casper Hansen, who's one of our other neuroscience faculty, studies. And he's kind of a big deal in the NMDA receptor field, honestly. So yes, if this excites you, you should definitely reach out to him and ask if he's taken students. Okay, so for the NMDA receptor, this is a nice cross section of the NMDA receptor showing us different sites that we have previously discussed. We have our glutamatergic binding site, we have our glycine binding site, and then here is our magnesium binding site. Those are probably the three most important sites for you on this slide. With that being said, the things that are shown in green are drugs that activate this receptor. So things like NMDA, lysine, D-serine, all are going to bind to the site and activate them. They act as agonists. And then anything shown in red are going to act as inhibitors for this receptor. So D-aminophosphonate, which is also known as AP5, blocks the NMDA receptor. And then magnesium is also obviously going to block the receptor. And then interestingly, you'll also see things like a PCP binding site, right? You'll notice that as a drug. And you can see phenylcyclidine binds to that PCP binding site, as does ketamine, which is probably something that you've been hearing more and more about in kind of the common vernacular. The purpose, or what ketamine does, is it blocks the NMDA receptor. It binds this PCP binding site, which you'll notice is actually kind of under this magnesium binding site, which means that things that bind to this PCP binding site only bind to receptors that have been activated. We call this use-dependent blocking, meaning that only once an NMDA receptor is used does that particular site on the receptor become accessible to these drugs. So that's true for ketamine, and it's also true for another drug called MK801. Kind of, yeah.
Oh, sorry, this goes back.
Yeah.
You just cover how calcium needs to long-term nutrition.
Ooh.
Or is that?
No. It's a good question. It's definitely a good question. So, so calcium is kind of one of those linchpin signaling molecules that's required for long-term potentiation. But keep in mind, there's a lot of different ways in which our cells kind of have access or allow calcium to flux into their cells. We will be talking about like long-term potentiation as a molecular process and memory as a learning process for literally the first month of the spring semester. So we will discuss long-term potentiation in that regards. Calcium levels, it's a delicate process, right? We, I'm sorry, I asked this question. So based off of NMDA receptor composition can determine how permeable a receptor is to calcium. The very permeable ones can facilitate long-term potentiation, and the ones that aren't as permeable actually cause long-term depression, which is kind of a retreating of those dendritic spines and that kind of conversation that happens between those synapses. So depending on the NMDA receptor concentration will determine whether you get more plasticity and more potentiation and more kind of long-term crosstalk of those two synapses. And then in the case of the other receptor subtype, you'll actually just kind of be tickling, giving low concentrations of calcium, and that actually depresses that conversation that those two synapses are having. Now, I know that's a pretty superficial response because we're not talking about all of the downstream signaling pathways in which calcium facilitates the potentiation. I'm getting kind of auctioneering here, so I just have to take a breath. Short-term potentiation is caused by almost immediate receptor expression onto the postsynaptic cell. That's how the cell kind of like short-term responds. But then you can also cause long-term genetic changes depending on how long that calcium change occurs within that cell too. So then you actually get, transcription factors that become upregulated and cause for more protein production and more postsynaptic density to be created in that instance. Does that help? Okay, sorry. I'm happy to go off on that, but I also don't want to get you guys really overwhelmed to be like, do we need to know that for the exam? No, not right now. We will get there though, I promise. And that again is probably one of the more interesting components to neural plasticity is understanding how those connections are made, when they're made, and what types of receptors facilitate it. NMDA receptor dependent plasticity or learning and memory is very well characterized in several different areas of our brain. One area that gets characterized a lot is the area called the hippocampus, because that's our area that's important for memory consolidation. And if you, and I've done this experiment, if you apply this D-aminophospho, this AP5, you can block all long-term memory and learning in that area of the brain. It's impressive actually. Where you normally would get this huge increase in synaptic plasticity, it's just a flat line. So it just kind of depends on the type of learning and memory that you're characterizing, where you're characterizing it at, and the receptor subtypes that are facilitating it. Okay, so yes, glutamate is, did you want to say something else?
No, I'll ask you after class.
Okay, that's fine. So glutamate sounds like a great thing, right? It's great that you have this like ability to have this excitatory neurotransmission that's occurring in the brain. However, it's a delicate balance. Too much glutamate signaling actually harms our cells and can lead to things like cellular death. So during periods of things like ischemia, what is ischemia? Yeah. Ischemia is not a symptom of stroke. It's an effect of stroke, right? So if you have a stroke, apparently I'm having one right now. No, so if you have a stroke, whenever I teach about strokes and in my other courses, I invariably get like a terrible headache the next day and I think I'm having one. It's just terrible. Anyway, so during stroke, depending on the type of stroke, you can lead to both oxygen and glucose deprivation, which we know as neurons they need. And so during ischemia, which again is that lack of oxygen and glucose, and hypoglycemia, which is a lack of glucose. So when you do not give the neurons what they need, you will see cellular death that occurs. When the cells die, you have unregulated / dysregulated glutamate being released all over the place. And so that what this does, it kind of overwhelms the reuptake mechanisms, and you get what we call excitotoxicity. Excitotoxicity is essentially as an excitement event that leads to toxic downstream effects of the cell. So this leads to an increase in intracellular levels of calcium, like as we were just talking about a second ago. A little bit of calcium is okay. This is kind of Goldilocks. You want to be just right, because too much calcium is going to activate all these downstream pathways, which leads to apoptotic signaling, right? And that's cellular death. So it just kind of becomes this exacerbating snowballing effect where if you have these cells die that lead to the really unregulated release of glutamate, it can actually bind to other cells and lead to excitotoxicity and a ton of calcium flexing into the cell and cell death. Yes.
So this would happen during like type 1 diabetic shock. Okay.
Yeah. So anytime again, when your neurons are not getting what they need, whether it be oxygen, glucose, or oxygen and glucose primarily, you are going to start to see cell death that occurs. So yes, you might see that happen during a diabetic event. Because of this fact, we are starting to look at antagonists or blockers for their therapeutic value. Like, are there ways for us during excitotoxic events to kind of dampen down these receptors that are allowing for calcium to come into the cell? Okay. So these are, again, focusing primarily on our ionotropic receptors, our AMPA and our NMDA. We also have our mGluRs, our metabotropic receptors. As you will quickly notice, is that this is highlighting all of the different places in which you see metabotropic glutamate receptors being expressed. They're just not on the postsynaptic cell. We also see them on the presynaptic GABAergic cell. We see them on the presynaptic glutamatergic cell. We even see a little bit on the astrocytes as well. So it gives you some insight as to like, yes, as somebody who's first learning neuroscience, you're just like, just tell me the simplest answer. Unfortunately, this is where, again, it gets kind of complex. And the fact that you have mGluRs expressed at kind of every different location within this kind of synaptic interaction, whether it be the postsynaptic cell or the presynaptic cell, or even on the astrocytes. This is kind of what current literature has shown us. The mGluRs, as we saw previously, are grouped into 3 groups, most often based upon their G-alpha coupling, which again, I have no interest in having you memorize pathways of the G-alpha systems. Just keep in mind, S and Q tend to be excitatory, I and O tend to be inhibitory. We'll just kind of keep it simplified like that. I and O, and again, one of the reasons that we have the group 2 and group 3 is that they are expressed on the presynaptic synapses. So they act as what we like to call autoreceptors. It's like a negative feedback inhibition. If there is too much glutamate in this synapse, it binds to group 2 or group 3 receptors, and that allows for less glutamate to be released. It's like a negative feedback loop. Our physiological systems love a negative feedback loop because when things kind of get out of whack, the binding to receptors like this kind of bring us back into check to make sure there's not too much glutamate present. Does that make sense? Hopefully. Yeah. Okay. Ooh, last one for glutamate. Last thing for glutamate. So as we were talking about glutamate, this slide, I don't need you to memorize like drugs, specific drugs and what disease states. I like to just introduce this slide because often when we're learning about neuroscience, it kind of seems a little bit removed from maybe like clinical applications. And this gives you some insight as to like how we actually target said systems. Now, with that being said, our glutamatergic system is found, as I said before, ubiquitously across all of our brain, right? So it's kind of a hard system to modulate, or that to kind of not deal with, but kind of modulate and not have a ton of adverse side effects because you're affecting every area of cognition when you either activate or block it. So first we have, again, NMDA receptor antagonists, things like ketamine. It can be used as a general anesthetic. Some individuals use it for that purpose. One of the reasons that they don't use it is because it induces hallucinations. So it does have that use as well as a hallucinogen. We are seeing it more and more utilized in kind of behavioral therapies or drug therapies for things like depression. And so that's kind of an emerging field, I would say, of ketamine use. Memantine also is an NMDA receptor antagonist. This is used for Alzheimer's disease. It's fairly what we call low affinity, meaning that it's not super sticky to the receptor, just kind of tickles it. But doesn't, because it just kind of tickles the receptor, you're not going to inhibit kind of normal cognitive function. For AMPA receptor modulators, we can see here an antagonist used as an anticonvulsant for treating epilepsy. Epilepsy/seizures are dysregulated and overexcitation of our brain systems. And so it's not surprising to see here that one of our answers to that is to start to kind of block and diminish that excitatory action of the neurotransmitter system. Other glutamate release inhibitors, again, we've got one here, Rizulol, which is going to be a ALS treatment that decreases glutamate release and provides some neuroprotective effects for ALS patients. So these are just some drugs. Again, I don't need you to memorize drugs or what systems are used for. This particular slide is more just kind of an FYI, so you can tie together kind of the basic science that we're learning into some maybe interesting clinical applications of said information. Yeah.
Are these all applicable to adults and then intentionally not children because of the way that children's brains are completely different stages of development?
Yeah, so most of these, the safety profiles for all of these drugs were originally characterized in clinical trials with adults, right? And then usually for use in children, you'll see like things like that decrease in dosage, but then you also have to go through a similar clinical trial with that particular patient population. So I can't speak to, let's say, all if any or all of these have been approved for use in children. That would take like, yeah, just another extension, I guess, of my own research related to this. But because kids have this high degree of neuroplasticity, I'm going to guess they're contraindicated against the use in kids, just because you kind of don't want to interfere with that kind of normal trajectory of plasticity over time, because these receptors are so important to mediate those processes. Does that make sense?
That doesn't.
Yeah. Yeah. Okay. 50 minutes in. How's it going, everyone? It's a lot. Okay. Well, let's take 5. Okay. Let's take 5. I know I'm getting into it. This is dense. This is definitely neuroscience. This is definitely neuropharmacology. But let's take 5, stretch it out. We'll come back and start class again at 2:55, 2:57. If you need to go to the bathroom, go to the bathroom. If you just want to go outside and get a breath of fresh air, get a breath of fresh air. But let's just take 5. I recommend that everyone at least stand up. I don't care if you go anywhere, but just do a quick stretch so we can finish out strong.
No.
Yeah, I figured that.
Yeah, it's not.
Actually, yeah.
It's like my
shape.
Yeah, sorry. I should.
No, it's all right. It's okay. Go outside.
Hey, what's up?
I haven't thrown my car off at some appointment thing. Is there a class activity? Okay.
No,
My computer excited. It's a whole thing. But anyway,
you're good.
Okay.
thank
Go,
you.
go enjoy your life. I'm recording this.
Awesome. I'll see you Thursday.
Okay.
The gap. Okay, technically I'm thinking about like. No, because you have to get like, you can smell the boxes. Yeah, that's right. Yeah. So they will literally. They don't do it, but it's not a big jump up. Yeah, I mean, it's a whole lot, I think there's gonna be like. Yeah. Goes from 6 to 10 p.m. Yeah, I'm, what mile is that for you? That's the only lab I can do, so if you can get in, what time is that? And it's so late one on Thursday, from 6 to 10, because I actually cannot do any, unless I want to take it Friday at 8 a.m., but first I feel. Yeah. Like, I think I'd rather do 6 p.m. to 10 p.m. on Thursday night, but. I'll rearrange it, I think that's some huge shit. Okay. Cause yeah. I really don't want all 1 p.m. start and then a random paper. I'm saying, and then I actually said that the first place.
Boop.
One of my friends taking us for lunch, we're still offering. I don't know if we would just go next to it. Yeah, they do, which is kind of, just although. I think I'm taking 21. That's me literally every semester after this, beginning semester. Yeah. Or live in the library. I'm gonna create myself with the stairs. It's so there, yes, that's it. Yeah, no, that's how I'm starting to get outside, because I'm noticing that I'm studying this group stuff, and when I saw it, I do it, so now I have to pick up it. I'm just like, there's like, if I'm really productive one day, the next day, that's good. Like, you said that all of your. I know that I did, like, my words, I missed all of that. And then today I was like, I'm not. It's this study.
Reason.
You
grow toxin.
What is that? It's a very effective blocker of GABA.
Interesting.
Yeah. Thanks to the carbon cake, right?
Yeah, it's not labeled, it's a carbon.
Yay, organic chemistry.
It's gonna be like one chemistry, chemistry minor. It's gonna be like one thing of chemistry, it's like the one I've been.
I haven't taken chemistry. I think as long as we can get away with not taking it, I can keep that.
Oh, because you know,
Yeah.
well, I might not.
I've always been told that's that's like.
No, I was kind of mad, and that was my advisor the other day. He was like, it's a funny story, you have to take it 151.
Oh.
And I've already taken 121, and I'm in intro organic with him right now. So I'm pretty upset at him right now.
Is it Cooper?
Luke.
Oh, Luke Alfred.
Yeah.
We good?
What?
Are you headed out?
No.
Okay. Oh, okay.
It's stretching. I'm not just leaving.
Okay.
That's or like. I'm so tired.
I'm just and I'm like, hey, look, here's what I want to do, like, how can we achieve this, you know? How about this? I use
Right
that
after.
class, you need to take the.
Would you mind if I heated up my?
And I go, I can do that. And I was like, no, that's.
I have a feeling we're gonna be done sooner than.
That's why.
Okay, if you wanna take, I have another meeting at 4.
Yeah,
I think as long as we get there by like 3:40, we should be good. 20 minutes should be good. Yeah.
Like you go.
Okay, you guys ready to power through? We have all of, I think, 5 slides left. And then we'll take a break until Thursday. Okay. So we're still in GABA. Okay. So we talked about glutamate and now we're gonna talk about GABA and glycine. I'll just put your fears to rest. There will never be any chemical structures on any exam I give you. So do not worry, I'm never gonna give you a picture of glutamate and a picture of GABA and be like, good luck, tell me which one is which. No. I'm not overly concerned with that. But GABA is a inhibitory neurotransmitter. But if you did look at GABA glutamate side by side, it probably would be hard to discern them because they do look structurally very similar. This image here is just giving you some examples of, again, the inherent structure of both GABA and glycine. Glycine is incredibly small in terms of a structure. But you can see also additional kind of pharmacological agents that either can block or activate said receptor sometimes. Okay. Now, GABA is released from interneurons, primarily in the central nervous system and spinal cord. Glycine is primarily found in the spinal cord and the brainstem. Okay. We'll talk more about the receptor subtypes moving forward, but how GABA exerts its inhibitory action is through the influx of chloride through the ionotropic receptor. So let's start at our first step of this system, which is GABA synthesis. GABA-T transanimates alpha-Q glutarate to form glutamate and activates GABA to succinate semialdehyde, but it's the presence of this GAD enzyme here that converts glutamate to GABA. It's literally the presence of this one enzyme in the presynaptic cell that determines whether a cell is excitatory or inhibitory. Okay, and again, you might be like, that's fun, why do I care? Because if you were to take a slice of brain and you wanted to know which of these neurons is excitatory and which is inhibitory, what you can do is you could just use a stain that stained all neurons, and then you could just co-stain for, let's say, that something that highlights GAD, and your GAD positive neurons are going to be your inhibitory interneurons. So it allows you to kind of physically look at a slice of brain and say, this is an excitatory neuron and this is an inhibitory one. So this presence of the GAD again helps kind of determine the difference between a glutamatergic and a GABAergic cell. Now, again, in terms of these pathways, I don't need you to get into the weeds. We just spent some time talking about GAD. That's probably worth noting. But in terms of all of the other enzymes that are presented here, don't get too bogged down in them. So we have GABA. It's going to get loaded into our presynaptic vesicle, which is called VGAT. Again, this type of nomenclature is pretty helpful. We're going to go to the next slide. So that VGAT loads the GABA into that presynaptic vesicle. Once it action potential arrives, it gets released and it hits a series of receptors. And now, luckily, GABA is a little bit more straightforward. We have your GABA A receptor, which is ionotropic, and your GABA B receptor, which is your G protein or metabotropic receptor. So the way that the signaling in the system is going to be terminated is through the GAT transporter. This is kind of, again, the little vacuum that sucks up that neurotransmitter and stops it from signaling. And so the GAT, this GAT system is the one that stops neurotransmission in the GABAergic synapses. We have 4 different subtypes of GATs on neurons and glia that act as kind of little neurotransmitter vacuums and suck up that GABA and stop it from signaling. Again, if GAT transport is your thing, Dr. Mike Kavanaugh studies that as one of our researchers here on campus, so you can study GAT with him. Yes?
What is GAS?
I think it's GABAergic amino acid transporter, but that's a guess. But you're catching me on this one. I don't know. We'll have to like, yeah, well, it makes sense, but is it true? I will get back to you. Do not put that on an exam. Do it. I'm sure it's gamma aminobutyric acid transporter, and that's probably what it is. But so as we talk more about the receptors that GABA binds to, we have GABA A, which is ionotropic. It's fast neurotransmission. It's pentameric, meaning it has 5 different subunits, penta meaning 5, and it is chloride selective, okay? So earlier, when we were talking about depolarization, again, that's sodium fluxing into the cell, GABA A, it can facilitate hyperpolarization, or the cell becoming more negative through the influx of chloride. This is pharmacologically quite important, as we will see here in a couple slides. GABA B receptors are metabotropic. They are heterodimers. They're coupled to G-alpha I and O. They either inhibit calcium or activate potassium channels. If you activate a potassium channel, what's going to happen? If you open a potassium channel, Which way does potassium want to go? Out or in? Wants to go outside of the cell, right? Based off of that concentration gradient and that salty banana that we love so much, right? So potassium is going to leave the cell and it's going to cause the cell to become more negative. Now, GABA B is going to be found in perisynaptic regions and be activated by what we like to call GABA spillover once it kind of leaves the synapse. And again, also you see there's an agonist such as muscle relaxants or GHB, which is considered to be a psychoactive drug. Okay, now, what is GABA? Why is it important? Why am I going to highlight it as a drug target? Oh my goodness, how many things bind to GABA? GABA A, the ionotropic receptor. Benzodiazepines are what we call positive allosteric modulators of the GABA A receptor. When I say positive allosteric modulators, that means that these drugs alter either opening time or opening frequency of said receptor, but do not bind to the GABA site. The only way positive allosteric modulators can exert their effects is in the presence of GABA, okay? So it's kind of like you have to have GABA to open up the door, but then these positive allosteric modulators are going to amplify the effect that door opening has. So benzodiazepines act through that action. Barbiturates, ethanol. This is one reason that if somebody is consuming a benzo, do not allow them to consume alcohol because you're modifying the same receptor. You're amplifying that inhibitory effect, which is again the reason you get sedation and can have coma induction in that instance. They're used to treat neuropsychiatric disorders, epilepsy, Huntington's, addictions, sleep disorders. They can be used as general anesthetics, as hypnotics, meaning sleep inducers. It's impressive as to how many different ways we can modulate the GABA A receptor to inhibit our brain's action. Okay. Questions?
If someone were to use like benzodiazepines mixed with alcohol when they were younger, like prior to their brain fully developing, could that affect like the way that their brain is taking in signals for the rest of their life?
That's a ton. I guess it depends on how extended that particular combination was utilized, right? So like an acute exposure might not have the same type of long-lasting effects as chronic continuous exposure, right? Then you're kind of continuously re-altering the signaling pathways that might otherwise form during kind of, we'll say, quote unquote, like normal neuroplasticity events. However, with that being said, when we talk about neuroplasticity, that's one of the beautiful things about our brains is that we can retrain them and we can kind of reinforce certain pathways that developed. You know, like there are many fields, supportive fields in cognitive development that can help mitigate, let's say, any level of deficit that may have occurred earlier developmentally, right? Like I'm thinking about like speech and occupational therapy that allows some of those pathways to kind of reform in those specific ways. So yes, there might be some deficit that can be incurred, especially if it's like a chronic exposure. However, with like certain levels of plasticity and other events to support that retraining of the brain, that's one of the ways in which you can kind of rewire it. So that's like the amazing thing about the brain is like you always have this capacity to kind of maybe rehabilitate pathways that were previously lost. Like you see that with stroke patients, you see that with maybe even some like Alzheimer's patients, you know, just a little bit of like reformation of specific pathways if you kind of treat them the right way. Does that help?
Yeah, and do you think if there was continuous exposure that could change their personality to kind of like mimic symptoms of mental health disorders?
Absolutely. I would say yes, because you're kind of chronically inhibiting areas of the brain that maybe needed to be active at that particular like neurodevelopmental phase. So yes, it could manifest in that way because the GABAergic system is kind of, as we were talking about before, is found ubiquitously across the brain, even areas of our brain that do facilitate things like personality development and thinking specifically of like our frontal cortex, right? So if you're taking a benzo and you're also consuming ethanol, that's going to be hitting your frontal cortex as much as it's hitting every other area of your brain. And that's what's going to facilitate things like personality development and possible mitigation of like mood disorders too that could arise. Yeah. Okay.
You're right.
I was right?
Yes.
Yay, what was the actual answer? Gamma aminobutyric acid transporter? Woo!
You're right, got it.
I don't know about that. Are you serious?
Oh, I was just a little bit like curious about how it works in sleep disorders.
Yeah, so with sleep disorders, the way that this is used is just basically as a sedative. So there's almost like a concentration dependent effect of inhibition, right? You just give a low concentration of, let's say, a benzodiazepine or even a barbiturate, and that's gonna like, so a small dosage of that is gonna be a sedative, but high enough doses are gonna lead to things like anesthetic effects, like make it act more like a general anesthetic, and then at high enough concentrations, you're going to get coma induction. So it's like a concentration dependent response, if that makes sense.
Does somebody then go through normal stages of sleep? Like, are they actually receiving like REM sleep?
No, usually sleep architecture is disrupted with most hypnotics that are consumed. And in a, sorry, folks, I just lectured on hypnotics like 2 weeks ago, but not in this class, obviously, but if a perfect hypnotic is going to rapidly put you to sleep, keep you asleep, and allow you to follow normal sleep architecture. But there are very few hypnotics that allow for that, all of those processes to occur. So when you again modulate things like the GABA receptor, you usually will get quick sleep induction. You might even be able to stay asleep, but you're not going to get as many REM cycles as you otherwise would see across kind of the sleep span of that particular like sleep cycle or what have you. Yeah, but we're also starting to look at other receptors. Obviously, things like melatonin could be one of those things that you could possibly modulate. There are some serotonin receptors that are important for sleep as well. So right now, pharmacologically, we're trying to identify other neurotransmitter systems that might be more important for sleep induction and can help us adhere to more normal sleep architecture. We obviously also use antihistamines for that purpose as well. Other questions? 309? Okay. Okay. So before I let you go today, think about any questions you might have. I know today's lecture was pretty dense. We have covered a lot of ground, but hopefully it feels like, hey, I'm really in neuroscience class now. I'm learning about neurotransmitters. On Thursday, we are gonna discuss acetylcholine, and then I will give you an example, kind of example of maybe I'll make the example in-class activity related to this material. So you guys can see a little bit of what that end of semester project is going to look like. And then we can also reinforce some concepts that we went over today, as well as on Thursday. So our in-class activity will be on Thursday, and you will actually be doing something in class. So come, spread the word, tell folks who aren't in class to come to class. But in the meantime, I will continue to work on grading your exams. If you have any questions, concerns about today's lecture, about the in-class activity, about the semester-long project, feel free to reach out. If you have questions about career trajectories, anything like that, feel free to reach out. If you have questions about what 281 is going to look like, because I know many of you are registering right now, is that correct?
Yeah.
So if you have questions about 281 and what that's going to look like, feel free to ask me. Keep in mind when I talk about 280 is kind of basic science. That's where we're at here. 281, learning and memory. We'll talk about all of the sensory systems. We'll talk about complex cognitive processes, the difference between emotions and feelings. So maybe one of the reasons that you got into this class originally is something we cover in 281, hate to tell you. But if you have questions about that too, I'm happy to share last year's syllabus with you as well. Okay, that's it. I will let you go. Enjoy your lives. Please make sure you're taking care of yourselves, getting plenty to sleep, eat, all that other stuff, okay? I'll see you all on Thursday.
I knew